Hello everyone, welcome to this live session. So I'm Gaëtan Poder from Alliance Bioexpertise company. We are a French company located to the south of Rennes. We develop and manufacture lab equipments for microbiology laboratory, but also cell culture laboratory. And also we distribute lab equipments for cell culture and mainly Ruskin. So today I'm with Petra Mekulainen from Ruskin company. Petra, can you introduce yourself?
Yes, thank you, Gaëtan, and good afternoon also from my behalf. So I am Petra Mekulainen, and I am the sales manager at Baker Ruskin in Europe. So Baker Ruskin is part of the Baker company, headquartered in Stanford, Maine, in the US. And the Baker activities, the Ruskin activities are located in UK. And our sibling company, Clean Air by Baker, is located in the Netherlands. So we are the world leaders in contamination control and precision atmospheres for life sciences and for research laboratories. And we have been actually the first ones to develop these products. So Ruskin developed the first commercially available Hypoxia Workstation over 20 years ago. So we have the absolute expertise in this field. And not only that we manufacture these products, but we have always been supporting science and scientists. And over the years have had great collaborations with the leading scientists and got to learn about the needs of the field. So our advanced range of closed cell culture workstation helps researchers to get more reliable and more translational results by using the controlled atmosphere and the physiological oxygen levels.
Thank you, Petra, for this presentation. Can you tell us a bit more about what do you mean by physiological cell culture conditions?
Yes, so by physiological oxygen level, I mean the oxygen concentration that is relevant in the tissue of origin. So the oxygen concentration varies between different tissues and cells. And most of the human tissues, the range is from about 2% up to 10%. So actually, the cellular functions they occur in quite a narrow oxygen range. So, for example, the lung epithelial cells they reside in approximately 14% of oxygen, which is among the highest in the human body. Compared to then, for example, brain cells that function in much lower oxygen condition, about 2 to 3% only. And this is kind of so to say that exposing these cells to anything above or below their natural environment will have some consequences that are basically impossible to track.
As you say, you want to be as stable as possible. So how do researchers work on cell culture in the lab mainly?
Yes, so the most of the cell culture is done, so to say, on an open bench. Yeah. So there, the manipulation and handling of the cells will happen in a biological safety cabinet, yes. And the cells are incubated in a CO2 controlled incubator. And in most of the labs, these incubators they are opened and closed many times a day, which causes then fluctuations to the environment where the cells are kept. And of course, so does taking the cells out for handling or for microscopy and so on. And basically, these fluctuations will eventually have a drastic effect on the behavior, on the expression patterns, everything in these cells in question.
Okay, so for example, if I work on cancer cell line, and I think cancer cell line like hypoxia.
Yes.
So how do practicing under a workstation will differ from my open bench practice?
Yes, it is especially important to consider the oxygen levels in cancer research because solid tumors are known to be very hypoxic by their nature, and that reflects to changes in the metabolism of these cancer cells. So our in vivo workstation here, so it represents the closed cell culture method. So the main idea is to minimize any fluctuations in the environment, keep the stable, controlled atmosphere inside, and also to keep the cells in their natural state. So the in vivo workstation, it kind of combines the biological safety cabinet and an incubator.
Okay, so that's nice. As you say, we really want to minimize all fluctuation in atmosphere. So can you explain how the workstation works actually?
Yes, so we have the internal chamber here, and the internal chamber is not only temperature and humidity controlled, but also controlled by the CO2 level and the oxygen level. And also we have an integrated HEPA filter in our Baker Ruskin workstations, and the internal atmosphere is constantly circulated through these HEPA filters to maintain the atmosphere clean.
Thank you. I have another specific question because you speak about control atmosphere. So how is the internal atmosphere control? We can control O2, CO2.
Yes. So here on the side we have a touch screen where the user can easily operate the workstation and set all the parameters. So, for example, the oxygen level that can be set from 0.1% up to 23%. And similarly, the CO2 level that can be set from 0.1%, but all the way up to 30% in 0.1% increments. And of course, the temperature and the humidity can be controlled as well. Yes.
Okay, perfect. So I spent some time doing cell culture, but many years ago now. So my question, if I remember well, we need sometimes to add some instruments to perform your cell culture. So can we add like a microcentrifuge or a microscope into this kind of workstation?
Yes. So the whole front screen here is removable, so you can actually easily take it off and place any instruments or equipment that you need on your cell culture workflow. So that can be, like you mentioned, a little centrifuge. It can be a microscope or imaging system. So many of our customers have had a great experience with the Etaluma LumaScope digital microscopes that are especially designed to be used inside such a workstation or an incubator. And that's a really compact system. It comes without oculars. So as you might see, there is a screen inside the workstation. So the what you see under the microscope will appear directly on the monitor inside the workstation.
That's really cool. But in my lab, I have a huge microscope right now. It will not fit into your workstation, but I really like to maintain my atmosphere for my cell culture. So how can I do?
Yes, I'm glad that you asked that, because that's of course what's the situation in most of the labs, that you have your big microscope in your cell culture that is used to monitor the cells. So we actually have a portable tool called Condocell. that is especially designed for the purpose that you just described. So it is a box. It has a glass bottom and a top, and it can retain the atmosphere inside for a short while. So what you would basically do is that you would take your condensel inside your workstation. You would be loading your cell culture flask or your well plate inside, and then you can take the thing out for a little checkup under the microscope. And after you're finished, you'll just put the materials back. Is it cells? It can be organoid cultures, whatever.
I think we have done some cell culture last night.
Yes.
Can you show us how to use this condensel and take out the cells from this in vivo 400?
Yes, of course. So let's open the interlock. So here on the side we have like a side port that is used to take any materials inside the workstation or outside. And I will be placing my condensel here, and when I'm closing the interlock, the interlock will purge, meaning that the ambient air is actually being replaced by nitrogen, so that we don't introduce any air inside the workstation, and it does not disturb the internal atmosphere. And now I am entering the workstation via these sleeves and the glove ports. So it is actually very easy. I'll just open the internal doors of the glove ports, and now I can open the interlock door and slide my condensel in. And like Tom mentioned, we have some cell culture dishes readily prepared, and I have placed now one dish inside the condensel, and I will be sealing it inside here. And now I'll put it back into the interlock, close it. And now I can exit the workstation. This quickly. Yes. And we can again open the interlock door to have our cells to be monitored under your big microscope.
Excellent. Thank you, Petra. I have another question in mind, because I know that you can control O2, CO2, but I think we need also to control maybe all humidity we have into this workstation. Can you give me a bit more details on how to do that?
Yes, so we have basically two options. So we have an optional accessory, an automatic humidity controlling system that is integrated into the workstation, which then would automatically humidify the internal atmosphere.
Okay.
The other option is to use these Aquasorb sachets. So it's a little sachet that contains sorbent material. The user will soak that in a sterile water and place it inside the workstation, where it then slowly releases the moisture into the atmosphere. So then you don't experience
bad experience.
Yeah.
Thank you, Petra. I really like this workstation, but I just begin to work on cell culture, so I don't have a really high budget. My question, I want to just do a few experiments at low oxygen conditions, and I have plenty of incubators into my laboratory. So do you have something that can fit my incubators just to test some low oxygen experience?
Yes, so we have actually just launched a new product called Foxbox, which is a smaller. It's kind of like an entry-level system, and exactly to the purpose that you were describing. So, conducting some short-term experiments in low oxygen level, some proof of principle studies, etc. So the Foxbox, it comprises of two parts. So here we have a cell culture chamber, and then we have a separate gas controlling unit. And the idea is that this cell culture chamber can be placed inside an incubator. The gas controller stays outside, but it controls the oxygen and the CO2 level inside the chamber. Yeah. So the chamber itself doesn't have it's not heated, so it will need the external incubator for the temperature control. Unless if you're working with some special application, for example, with some small model organisms, fruit flies, for example, so then you can also keep the cell culture chamber on the room temperature.
Nice.
And if you have an existing workstation, you can also place the cell culture chamber inside your workstation and thus create like an additional oxygen atmosphere.
Okay, so is that you can, for example, Normoxia into your Vivo station, and also you can directly switch to, if I can say, Physioxia. So, okay.
Yeah, yes. So this would be a perfect solution for your first Hypoxia experiments.
Perfect. And I think it's really easy to work with, because you just need to select your parameters and say go.
Yes, yes. So we have a very easy to use and intuitive software on the gas controller, like also in Vivo, but you'll just mark your set points for oxygen and for CO2, and then you press the play button and it starts to go down on the oxygen.
To equilibrate to what we want.
Yes.
Okay.
Yes.
Just to be sure if I understand everything well, what we can have as a workstation or Foxbox is really to have a stable atmosphere that will really stable and controlled atmosphere that will really be able to have an atmosphere which is close to what we physiologically want, and that will really permit us to mimic the condition where the cell lives. So
yes, that is absolutely perfectly wrapping up this this introduction of the systems.
I have to my side a last question, because we have plenty of researchers working in Hypoxia, Physioxia, and my question is, sometimes we have plenty of questions from different laboratories, but you have some, as you say before at just the beginning of the presentation, you have discussing with plenty of scientists. Do you have some kind of network where they can directly exchange or have more information about Physioxia?
Yes, so like I mentioned, we at Baker Ruskin have always been in active communication with the scientists, and actually after this summer we will kick off a virtual Hypoxia community platform called Hypoxie EU. So this will be a scientific platform for all scientists who are working in a hypoxia field or are interested in low oxygen research or physiological oxygen research. So yeah, we welcome everybody to join this community, join the events and the webinars that we are organizing. So stay tuned for some news.